Maybe. Okay. Alrighty. So this went okay yesterday. I did this with the Wednesday section yesterday and it was fine. It's a little bit long, it's a little bit dry because we're doing some neuron stuff. But there are going to be times when I'm hopefully asking you to maybe contribute or answer a question. Definitely, if you have questions, ask those questions and you can ask them just out loud. You can ask them in the chat. I will try to monitor the chat. And we'll just do this as good as we can do this. All right, let me share my screen. Okay. All right, so first I just want to tell you some administrative things. So the Canvas page, I did update this for next week. So if you look at this week's page, we have just a reminder that we didn't meet on Monday or Tuesday for you all. So I think I had said this in the announcement. I think what I'm gonna do is try to keep us on track by usually I devote a day to doing a sheep brain dissection in the biolab, and that is a real just like free for all, not a free for all. There's some guidance. But you're in the lab, you're just sort of playing around with the brain and looking at the brain. And then the next day we do a formal debriefing and we talk about some things that I really want you to get out of that. And that dissection never lasts an entire class. So I think what I'm going to do to compensate for the missed Tuesday is just combine those and do a little bit more formal of a guided sheep brain dissection, and also work in the stuff that you will need to know about brain anatomy in there. So I think that should work okay. And then doing this online, I really debated about whether to do synchronous or asynchronous, and I just decided that for me, it's a lot more efficient just to get on at the same time that I would normally teach and just talk through the material. And that way I know it's beneficial for some of you. Some of you do want to be here as live as possible, ask questions, etc. And for those of you who couldn't, then they can go and they can watch the lecture later. So that's what I decided to do. All right, so the materials are up for this week, and then for next week, I am trying to stay on schedule in terms of just like keeping quizzes when they initially were, and so this will still work to have your third quiz due next Thursday, and that will coincide with material that we're talking about for next week as well. And again, there's nothing else real formal due for next week. So does anybody have any administrative questions, anything that you want to ask or talk about before we go back to the material? Okay. I don't even get your eyes looking up at me. This is the thing I hate most about Zoom. I don't get any facial feedback for like we're good or we have questions or so you just gotta like jump out if you do. For me, I'm just like talking to my computer for 75 minutes. So it's a little bit awkward. All right, so let's go back then to talking about neural communication. So let me um, go back. Actually, what I'm going to do is I'm going to go through the Prezi here. Um, okay, let me just zoom forward to where we. Okay. So when we left off last week when we were in person, we were talking about what we needed to understand how two cells communicate, right? So we have our presynaptic cell and we have our postsynaptic cell. And I told you this is really, it's a circular process. And so it's hard. I could start here, I could start here, I could start here, right? Where do you start? And so I just usually we'll start with talking about the postsynaptic cell and defining resting potential, and then talking about how the presynaptic cell releases neurotransmitters and what that does to the postsynaptic cell. And then we'll talk about the action potential and how that repeats the cycle and leads to a repetition there. So we had left off last class, we were talking about resting potential, right? And I defined resting potential as when you think about this postsynaptic cell, And for now, let's think about this area here, right? So the membrane potential of a cell could be very different depending on where on the neuron you measure. So it's not just like in general psych, right? We're like, oh, the postsynaptic cell is at resting potential. There's different membrane potentials depending on where you're measuring on the cell. For now, we're going to kind of focus in on the cell body and we're going to focus in on the dendrites. And while those actually are not usually at resting potential, they tend to fluctuate in their membrane potential. Resting potential is really the baseline that we use to talk about these things. So, um, for that reason, I like to start with resting potential and just establish that as a baseline where we said the inside of the cell is 70 millivolts more negative than the outside of the cell. And so remember, we were talking about this concept of membrane potential, where we said you can measure the voltage of the inside of the cell and compare it to the voltage of the outside of the cell, and that is your membrane potential. And so you're going to hear the term potential used quite a bit in terms of um, resting potential, action potential, postsynaptic potential. And so the membrane potential when the cell is at resting potential, this is what we mean. You measure the inside of the cell and it is 70 millivolts less or more negative than the outside of the cell. And so remember, we say that that is polarized. And then just to remind you, we left off by saying anything you do to increase the membrane potential is going to make that difference bigger. And anything you do to decrease the membrane potential is going to make that difference smaller. So before I continue on and talk about the next concepts, I just want to make sure, do we have any questions about that? Do we need to clarify anything about membrane potential, especially? I know resting potential may still be a little kind of iffy for you, but like the idea of membrane potential. Are we good with that? Okay, I'm not seeing anybody jump up, so we will move on. Okay, so to change the membrane potential, right? I said that if you want to increase the membrane potential, you have to make the difference between the inside and the outside bigger. And you might wonder, like, how do you do that, right? Or how do you make the difference between the inside and the outside smaller? And what you have to do is you have to get ions to move across the membrane because the cell membrane is surrounded by different ions that we'll talk about and that you read about. And those ions help to maintain the membrane potential of the cell, right? And tell us what it is. So there's two ways that ions can move, right? There's more than two ways, but there's two key ways that ions move, especially when we think about across the cell membrane. And one of the ways that ions move is we say that they diffuse down their concentration gradient. So every ion we're going to talk about has a specific concentration gradient where they tend to be in higher concentrations at one part of the cell compared to another, right? If you're thinking about the cell membrane, you might have them at higher concentrations outside of the cell or inside of the cell. But if you give an ion a chance, that ion will diffuse. It will move so that it will equally distribute itself in the environment. So if there's a whole bunch of ions at high concentration somewhere, and you give them the opportunity, they will diffuse down their gradient to places of low concentration until they are equally distributed. And I'm going to try again. I have no facial feedback here to know like if you're taking notes or if you're processing. So I'm just going to try to give some pauses to make sure people can write down and jump in with questions. I often tell people when we talk about diffusion that this is like if you have really smelly sneakers in a closet, right, and that closet door is closed, then those things are going to be contained, right? Like the olfactory molecules will be contained in that closet. But as soon as you open the door, they're going to start to diffuse and they're going to start to distribute throughout the room. And then everybody will be able to smell them until they are equally distributed, right? And then at some point, they'll sort of dissipate out because they are so equally distributed that you won't smell them anymore. All right, another way that ions move is ions move based on their electrical charge. So you've probably been reading about these ions. Many ions that we care about in this process are positively charged. So sodium is positively charged, potassium and calcium. Some are negatively charged. So chloride is negatively charged. And ions that have charges will move toward areas that have the opposite charge as them. So this is called electrostatic pressure. So if I'm a positive ion, like a sodium ion, I will be attracted to negative environments and I will be repelled by, and I just noticed that that says negative charges repel. That should have said same charges repel. I will fix that on the Prezi. I am repelled by like charges, right? So two positives are going to push away. And this is sort of like, magnets, if you've ever tried to take magnets and push them together, similar poles, that's when you get these like you can't push the magnets together, but opposite poles will attract very easily. This is the same thing, right? So opposite charges will attract. Okay. Any questions about those two? Alrighty. Okay, so then let's talk a little bit about at resting potential and talk a little bit about these ions and what these ions look like. So at resting potential, there are four key ions that we are really interested in thinking about in terms of neuron communication. And those four ions are sodium, which is Na plus, potassium, K plus, chloride, not chlorine, but chloride, which is Cl minus negative, and then calcium. So those are the four ions that we are going to be interested in. Those ions are distributed across the cell membrane. So if you look at this picture here, what you're seeing is you're seeing the cell membrane. And if you remember in the classroom, we use the wall as an analogy, right? So we're inside the classroom and there's this big wall, and things can't get in and out of that wall, right? That wall is a barrier between the inside of the classroom and the hallway, right? The outside. And then embedded within the cell membrane, you have these proteins, and these proteins do different things. So we'll come back to this protein later on, but a lot of these proteins serve as gates. They serve as channels for ions to get into and out of the cell. So just like the door in the hallway wall that we have. And different ions have different distributions across the cell membrane, especially at resting potential. So we're going to talk right now, just like what does this look like at resting potential? So we are at one place on the cell. So let's just say like I'm a neuron, right? Remember? And I have my axon. So let's say that we're just talking about like this part right here of the cell. We assume it's at resting potential. What does the ion distribution look like? And so what this is telling you is it's telling you the absolute values. You do not need to know the absolute values, but I want you to pay attention to the relative values, right? So for like sodium, where is it higher? Is it inside or outside? What about potassium? What about chloride? And then one thing that textbooks often don't do a good job talking about is they don't do a good job talking about these negatively charged proteins. So I like that this image has these pictures on here. So these proteins are represented by black dots. They are not ions. They are actual structural proteins that are in the cell. And they tend to be more numerous in the cell and they carry a negative charge. And so when we talk about resting potential being negative 70 millivolts, that is largely maintained by these negatively charged proteins. Because if you look at this, like most of these ions are positive. So you have to ask, how does the cell have a negative charge if most of these are positive? And the only negative ion that we're talking about is chloride, and chloride is actually in higher concentration outside of the cell, right? So, like, why would the inside of the cell be negative 70? And the reason is because it just has these big negatively charged proteins that never leave. They just can't get out of the cell. They are too big to go through the membrane. They are not lipid soluble. They don't have channels that they can go through. They don't work with that door. And so they just hang out inside the cell. And so any change in membrane potential is a function of these ions moving across the membrane in relationship to these negatively charged proteins, and that will change the ion potential or the membrane potential. Okay, so let's start with sodium. Sodium is the easiest. Sodium is also one of the most important, right? When we think about uh, neuron communication. And so sodium as an ion is at higher concentration outside of the cell at rest. If we go back to this picture, right? And we look, sodium is represented by these blue dots. There's much more sodium on the outside of the cell than the inside of the cell. So if it's resting potential, you can assume that there is more sodium outside of the cell than inside. That is just something that's a given. That's just something that you want to, like, if you're in biological, anything biology, neuron communication, you just want to like know that sodium is at higher concentration outside of the cell at rest. Okay. And then sodium can't get through the cell membrane on its own. So it's not able, it's carrying that charge. So anything that's charged has a really hard time getting through that cell membrane. So sodium has to go through the door, right? It has to have a channel that it can go through. And so that's represented by this protein here. At resting potential, most of the sodium channels are closed. So these are not open doors for sodium to go in freely. Sodium is hanging out outside of the cell and it can't get in because the doors are closed. One of the things that we will talk about is there are ways to open these doors. And one of the ways that you can open the door is a neurotransmitter, right? We already talked about neurotransmitters. They fit into receptors, which are like key and lock, right? So if a neurotransmitter comes and fits into a receptor on the sodium channel, it can open the door. And so here's where I'm going to ask you if you're willing. And I know it's really hard because you can't see like who's participating. You just got to go like, say something and maybe talk over somebody else, right? And figure it out. But I'm going to ask you, what happens if I open that sodium door at resting potential, and why does that happen? So what does sodium do if I open that door? Anybody willing to guess? Take a stab.
Wouldn't it just go to like so it's equal at both sides?
Yeah, sodium's going to enter the cell, right? And what Abby just said is that because it diffuses, right? We talked about one of the reasons that an ion is going to move is because it wants to equally distribute, right, itself, and it moves down its concentration gradient. So we said that sodium is at higher concentration outside of the cell. We open the door. We open the channel, and so that means sodium is going to have a pressure to diffuse into the cell because it's like, well, I've been at high concentration. And now I want to go to a place of low concentration. All right, what's another reason? There's a second reason sodium wants to go into the cell. And I always say want, right? Like ions and cells don't have consciousness, free will. But what else is going to pressure sodium to go into the cell? What else do we know about ions moving?
The charge is more negative inside, so the sodium goes towards it.
Right? That electrostatic pressure. So the charge is more negative on the inside. And that means that sodium is going to be attracted to the negative inside of the cell. So this is just, and this is going to be a huge thing. When we talk about action potential, this is going to be really big. That sodium at rest is in high concentration outside of the cell. But if you open a sodium channel, sodium is going to have a lot of pressure to go into the cell, right? Because it's going to want to diffuse and it's going to be pulled in by the negative charge. This is, I can't remember if I used this analogy with you before, but I always tell students, so these situations where sometimes you're like taking a test and you might take a little bit longer than the class. And so you're kind of finishing up. And meanwhile, the next class is sort of building up outside the classroom, right? And so more and more people are standing out in the hallway, but the professor has the door closed because the professor's trying to let you finish up. And at some point you finish, as soon as the professor opens the door, it's like this just onslaught, right? Like all of these students come into the room. That is very much like sodium coming into the cell, right? There's just like all of this pressure to have this ion that built up in concentration to come into this environment. And so you'll get a lot of sodium coming into the cell. All right. I'll pause for a second. Ask me questions about this. Sometimes I lose my chat. Okay, there it is. Okay. Then let's talk about potassium. So potassium, slightly different. Also very, very important ion in terms of neuron communication. Potassium is different than sodium. Potassium is at higher concentration inside of the cell. So if you look here, right, potassium is these yellow dots, and these yellow dots are highly concentrated on the inside of the cell. When you think about the four key ions, sodium, potassium, chloride, and calcium, potassium is actually the only one that is in higher concentration inside the cell. So this is another good just like basis, right, to just have in terms of bio. Just know if we're thinking about high concentration of ions at rest inside the cell, it's going to be potassium. Okay. So, and here you can see again, they're showing you this protein. Sometimes those proteins, the negatively charged ones, are represented by an A. So just so you know that. All right, so potassium channels are a little bit different than sodium channels. Sodium channels, for the most part, we say are closed at resting potential. Potassium channels, many of them are closed at resting potential, but there's a few leaky ones. So this is why I said potassium channels are closed mainly at rest. There are a few that are open where potassium could, if the conditions are right, cross the cell membrane through those open channels. So just remember that there's some leaky channels. Okay. So again, if we're thinking about ion movement, right, and we're thinking about pressures for potassium to move, if potassium were able to, what would the diffusion pressure, um, what would that look like in terms of potassium? How would potassium want to move if you're thinking about the concentration gradient or the diffusion pressure?
Potassium would want to leave.
Right, because it's higher concentration on the inside, right? So like if it has the ability to go out of channels, yeah, it's going to want to diffuse out of the cell because it's going to want to come from high to low concentration. But if you think about the electrostatic pressure, what does the electrostatic pressure predict? It's going to want to leave. It's going to want to stay in the cell. Trying to wait out the awkward silence. Oh, I see a chat. Somebody's answering. So Kennedy says it might want to leave, right? And that's what you would think in terms of thinking about sodium, right? Because the pressures were consistent. But remember that potassium is positive and the inside of the cell is negative. And so this is a little bit confusing with potassium in relationship to sodium. Sodium is pretty easy, but with potassium, the two forces are opposing. So the inside of the cell, sorry, the inside of the cell is actually going to try to hold the potassium in. So the two forces are opposite of each other. Diffusion predicts that it would go out, but electrostatic pressure says, well, I'm negative on the inside of the cell because we're at resting potential. So I'm going to hold the positive potassium in. All right, so that sets up this situation where at rest, potassium doesn't tend to move across the cell membrane all that much, even though it has the possibility in some of those leaky channels. So -70 millivolts is actually close to, and I know if you're a bio person, you know that this is actually, it's -90 and it's a little bit complex, but let's just say -70 is what we call potassium's equilibrium, electrochemical equilibrium. It is a place where the force of the electrostatic pressure is equal to the force of the concentration gradient, and thus potassium will not move, right? So at resting potential, even though there's a couple of leaky potassium channels, potassium is not going to go out those channels because the force of its wanting to diffuse out is counteracted by the force of its coming in, right? The electrostatic pressure holding it in. And so if we had more people and if we were in class and I could look at you, I would ask you like what needs to happen then to get potassium to move, right? What circumstances will get potassium to move? And so you have to change that equilibrium potential, right? You have to either open more potassium channels or you have to do something like, let's say some sodium got into the cell somehow, right? And we'll talk about how that could happen. Then this is no longer 70, right? Negative 70. If more positive ions have come into the cell via sodium, now we might be at like negative 65. That is no longer the equilibrium that potassium likes, and thus potassium will try to leave some of these channels to try to compensate and bring it back to its equilibrium, right? So as soon as you disrupt the potential, potassium, all of a sudden, like the diffusion pressure, the concentration gradient will outweigh the force of the electrostatic pressure, and some potassium will diffuse out. So if something like sodium enters the cell, potassium is going to exit to try to restore that equilibrium. I'm going to let you process that for a second. What questions do we have about that? What clarifications do we need to make for that? I'm going to take your silence as nothing for now. So this is actually, those negatively charged proteins we say are important for the resting potential, right? The reason that the inside is negative. But potassium is really important for maintaining resting potential because potassium is the thing that has the ability to compensate a little bit. And so if any change to the cell membrane potential happens, potassium will try to make adjustments by leaving the cell to restore it back to equilibrium. So it's a thing that actually really helps to keep the resting potential. Okay, so the next ion is chloride. Chloride actually is not all that important for the action potential and getting two neurons to communicate, but it's actually really important for inhibition. So we talked about excitatory neurotransmitters and we talked about inhibition. Chloride is really important for inhibition. So chloride is again at higher concentration outside of the cell membrane. So we have this higher number of green dots here. And so again, chloride channels at rest are closed. So chloride is not moving across the cell membrane if you are at rest with those channels closed. But if you are able to open a chloride channel, chloride is going to diffuse into the cell because chloride has this strong pressure, right, of the concentration gradient to go from higher to lower concentration. And chloride is complex in just like potassium because the diffusion pressure and the electrostatic pressure are opposing. So chloride you would think would have a hard time going into the cell because the inside of the cell is negative and chloride is negative, which means that they repel, right? Like it should not be able to enter the cell. And so chloride is very complex. There's a lot of things going on. If you're a bio person, you might know like sometimes the membrane just around these channels might be a little bit what we call depolarized, a little bit more positive, which makes it easier for chloride to get in and reduces that electrostatic pressure. But for the sake of our conversation, we are just going to accept that if a chloride channel opens, chloride will come into the cell because the concentration gradient is a stronger pressure than the electrostatic pressure. And so we're just going to say it's not like potassium where those two things equal out at rest. If you open a chloride channel, the force of wanting to come in because of concentration gradient is a little bit stronger than the force of electrostatic pressure keeping it out. And so the concentration gradient wins, and chloride will go into the cell. Any questions about that? Okay. And then our last item is calcium. Calcium is going to be really important next class when we talk about how we release neurotransmitters and have the presynaptic cell release those neurotransmitters. So calcium, again, higher concentration outside of the cell. The channels are closed. And this again is very similar to sodium, right? Because calcium is positively charged. So both the diffusion pressure and the electrostatic pressure are going to predict that calcium comes into the cell. So calcium very easily enters into the cell. There's no complexity about that. There's no opposing force. There's no equilibrium that we have to think about. Okay. So this is our baseline. This is our setup. When we think about resting potential, this is where we're starting. Everything is relative to this, right? Everything is in relation to this baseline that we've established. So I'm just going to look because I can't remember where I'm going next. Oh, yes, we're going to the synapse. So let me just give you a second and ask, what do we need to talk about? What do we need to repeat? What do we need to clarify to make sure that we're all on the same page with this? We feeling good? Give me some thumbs up. I can see some of your screens, so give me some thumbs up if we're good to move ahead. Okay, I'm getting some thumbs up. Okay. All righty. Some people are still there. It's exciting. Okay. All right, so the next thing that we're going to do then, so we established that this cell is at resting potential, right? And again, the entire cell is not at resting potential. That's really a misnomer. That's not a correct way of saying it. But we've talked about resting potential in the postsynaptic cell. So the next thing that we're going to do is we're going to zoom in and ask what happens at the level of the synaptic gap or the synaptic cleft. And so basically we're zooming into this little square here to talk about our next thing. And this is what that little square looks like close up. So here we have our presynaptic cells. So if you remember our candy activity, right, you held your candy in the left hand and you had your fingers, right? So think about like each of these being a terminal button, right, that has these vesicles, that has your candy that holds the neurotransmitters. And so what's going to happen is you release the neurotransmitters into the synaptic gap. Sometimes you hear this called the synaptic cleft, and they cross the gap and they bind to receptors on channels on the postsynaptic cell. Neurons, with the rare exception of some things we're not going to talk about, but like if you know about a gap junction or something, do not actually like touch each other. They do not have physical communication with each other. They communicate across these gaps. So even though we have like these billions of neurons in this network, none of them touch each other to communicate, right? It's all about communicating across this space. And another thing I wanted to tell you just about language is people often say like the synapse, right? So we're going to zoom in on the synapse. Technically, synapse is a verb to synapse. So it's the thing that is happening. And that space in between is called the synaptic gap or the synaptic cleft. But often people will refer to this as the synapse. And so just so you know that language. So the first thing I want to do in the synapse is we're going to talk about these receptors. And this is, again, if you're talking to the postsynaptic cell, right, this was the dendrites that somebody receive the neurotransmitters with. This is the key, or sorry, the lock that the key goes into. And we're just going to talk a little bit about different receptor types because these receptors can be slightly different. So the most common type of receptors that we talk about, not most common that exist, but the most common that we tend to teach about are called ionotropic receptors. These are the receptors that allow the ions to quickly go in or out of the cell through these channels. And so that's why it's called ionotropic. And so structurally, what these receptors look like, and this is a picture of these receptors within the cell membrane, they are formed out of multiple protein units, right? So we make these proteins, we synthesize these proteins, and they form into these receptor types. These proteins, so I often use the analogy of like my sports teams in high school. We used to do this, right? We would all stand sort of shoulder to shoulder in a circle, and then we'd put like a camera at the bottom and take a picture of us looking down. And so that sort of shoulder to shoulder reminds me of the way that these proteins are situated, such that it forms this pore, right? It forms a channel in the middle. So if you think about those people standing shoulder to shoulder in a circle, there's going to be a gap in the middle. That's how the ions can get in and out. That's the opening to the door. And then the way that you can get the channel to open, right, or to close is something called a ligand can bind to that receptor. You will hear, I'll play videos. Sometimes people say ligand, sometimes people say ligand. I'm actually more of a ligand person, but I know that bio and chem, a lot of people say ligand there. So just to kind of be consistent with what you might be hearing in other places. A ligand is basically a chemical like a neurotransmitter, a hormone, a drug, right? Some type of chemical that is binding to the receptor that can open the pore. And if the pore opens, then you have the possibility of ion movement, right? So just like with the sodium that we talked about, if sodium is all in this high concentration outside of the cell and it can't diffuse in and it can't be pulled in because the door is closed, as soon as a ligand binds, opens the pore, then sodium has the possibility to diffuse in, and it will be pulled in by the electrostatic pressure. And when that happens, it changes the difference between the inside and the outside of the cell, right? It changes the membrane potential so that it's no longer -70. All right, stop there for a second. That was a lot of information. Again, let you process, ask me questions. The second type is called a metabotropic receptor. And metabotropic receptors are a little bit more complex. We won't talk about them quite as much in this class, but really important for other topics like psychopharmacology. They're called metabotropic because instead of directly allowing ions in and out of the cell, it's more of a metabolic process that happens to get a change in the cell. So some things that are different. So this is the metabolic receptor here. So they're always kind of represented by these blobs. They do not have a pore, so there's no place. If they do have a binding site, so a ligand can bind, and here they're saying that a neurotransmitter is binding. But there's no pore. There's no place for an ion to come into or out of. Instead, they have this G protein. So they have this protein that is coupled. So sometimes you'll hear this called a G coupled protein receptor. And when the ligand binds, the G protein will dissociate and it will travel in the cell and it will do things in the cell. So one thing it can do is it can actually open these these ion channel receptors, these ion channels from the inside and allow ions to go in and out. So this is going to be a little bit of a slower process from the ionotropic receptors. Instead of a neurotransmitter just binding and allowing ions in or out, right? The G protein, the neurotransmitter has to bind here, the G protein has to leave, it has to travel, and then it has to activate the cell from the inside so that the ions can travel in or out. So it's a slightly slower process. Something else that the G proteins can do, and again, this is going to be pretty beyond the scope of what we're talking about in this class, but those G proteins can also interact with what we call effector enzymes. So an effector enzyme is something where if the G protein interacts, there will be this cascade of things that happen in the cell. And if you're a bio person, I'll give you an example. One of them is like cyclic AMP, right? So CAMP, it can affect to make either more or less of the cyclic AMP. So that too is going to have a change to the cell, right? So the overall story for both of these things is a neurotransmitter binds, it's going to change the cell in some way, right? It's either going to just directly open ion channels that allow ions to cross the membrane and change the membrane potential. And here I just want to clarify, sometimes people get confused here. Right, there's a difference between the ligand, so this is the neurotransmitter that is binding, and the actual ions. So let's say this is sodium that are coming in and out of the cell, right? Those are different things and they're represented differently. So you could either have that very fast change, right, as a function of binding to ionotropic receptors, or you could have slower and more complex changes by binding to a metabotropic receptor and having a G protein go and open an ion channel or interact with an effector enzyme. Okay, stop there. Questions or clarifications about that? I actually have a little movie of this, and I know it's weird to show a movie during uh, a Zoom lecture, but I'm going to do it anyway because this is what I would have done in class. Just to warn you, two things. This movie, the subtitles are in Korean. They used to be in English. Now they are defaulted to Korean. So I think if you're watching this at home, you can change those. The second thing is it's going to get a little detailed about the role of the G protein. So it's going to talk a little bit about like the alpha and the beta subunits of this G protein. We do not need to know that. It's going to talk about GDP and GTP. We do not need to know that. I am showing this to you mainly so you can just get a big picture visualization of the difference between the ionotropic function and the metabotropic function.
The opening or closing of postsynaptic ion channels is accomplished in different ways by two broad families of receptor proteins, ionotropic and metabotropic receptors. Ionotropic receptors are multimers made up of at least 4 or 5 individual protein subunits, each of which contributes to the pore of the ion channel. Ionotropic receptors contain two functional domains, an extracellular site that binds neurotransmitters, and a membrane-spanning domain that forms an ion channel. When a transmitter or ligand binds to an ionotropic receptor, the channel becomes activated and ions can flow through the channel. Ionotropic receptors generally mediate rapid postsynaptic effects. The postsynaptic potentials arise within a millisecond or two of an action potential invading the presynaptic terminal and last for only a few tens of milliseconds or less. The second family of neurotransmitter receptors are the metabotropic receptors, so called because the eventual movement of ions through a channel depends on one or more metabolic steps. Metabotropic receptors do not have ion channels as part of their structure. Instead, they affect other channels by the activation of intermediate molecules called G proteins. For this reason, metabotropic receptors are also called G protein coupled receptors. Metabotropic receptors have an extracellular domain that contains a neurotransmitter binding site and an intracellular domain that binds to G proteins. The G protein shown here has 3 subunits, alpha, beta, and gamma. The alpha subunit binds to guanine nucleotides, either GTP or GDP. Binding of GDP allows the alpha subunit to bind to the beta and gamma subunits to form an inactive trimer. Binding of an extracellular signal activates the metabotropic receptor and causes GDP to be replaced with GTP on the alpha subunit of the G protein. When GTP binds, the G protein becomes activated and the alpha subunit dissociates from the beta-gamma complex. Following activation, both the GTP-bound alpha subunit and the free beta-gamma complex can bind to downstream effector molecules that mediate a variety of responses in the target cell. For example, the G protein may interact directly with ion channels, causing the channels to open or close. Activated G proteins may also bind to other effector proteins, such as enzymes, that make intracellular messengers that mediate a variety of responses in the target cell. The activation of metabotropic receptors typically produces much slower responses, ranging from hundreds of milliseconds to minutes or even longer. A given transmitter may activate both ionotropic and metabotropic receptors to produce both fast and slow postsynaptic potentials at the same synapse.
Okay, so I know it's a little bit dry. And again, we do not need to know all the details about the GDP and the GTP and the dissociation. But I did want you to have that visual, right, to see just what that looks like in terms of the G protein opening those channels, or what that looks like in terms of the G protein influencing effector enzymes. And this is something like a while ago, right, when we did that activity with the candy, I said, you give your candy to a postsynaptic cell and that changes the cell in some way, right? That does something to the cell in some way. This is what I mean by that, right? When you give your neurotransmitter and it binds to the postsynaptic cell, it opens ion channels, allowing ions to come in that can change the cell membrane of the cell, of the membrane potential of the cell, right? Or you have neurotransmitters bind to activate this G protein that is doing these internal functions that's going to change the cell in some way. Okay. What do we need before we move on? Any questions before we move on? Any thoughts before we move on? Okay, so we're moving on. All right. So the next thing that we want to talk about is, well, what happens, right? When you have a neurotransmitter that binds to these receptors, what is that change, right? And why is that change important? So we have talked about membrane potential. We have talked about resting potential. And so now we're going to talk about these postsynaptic potentials. Postsynaptic potentials are really small local changes in the membrane potential that occur right where that synaptic connection happened. And so up until now, we've really been talking about, right, like just the presynaptic cell communicating with the postsynaptic cell, which is fairly artificial, right? Like it's never just two cells that communicate. And even when it is just two cells and you narrow it down, you still have all of these different presynaptic terminals and the ability to have multiple synapses at the same time, right? And so what this image is showing is a little bit more realistic, although still not as complex as it would ever be, but it shows that neurons will converge, right? It's not just that one postsynaptic cell is getting information from one presynaptic cell, right? And this is where, if we're in the classroom, I often pick, and I cannot remember, right, like where you sit and who's here and et cetera. But let's say just like student A is in the middle seat of the classroom, right? It's not just that student B is going to be the one talking to them. Student A is going to get neurotransmitters from student B, student C, student D, right? And so many different neurons that are making different neurotransmitters are going to converge onto the postsynaptic cell. And so that's what this is showing here. And this is a good time to remind you, any one cell only makes one neurotransmitter type, right? So this cell here might only make glutamate, for example, but it can receive multiple neurotransmitters types, right? So multiple cells are converging and synapsing onto that postsynaptic cell, and there's going to be multiple postsynaptic potentials. And every time there's neurotransmitter release and that influences the cell, it opens ion channels, right? Or it does something to those receptors, you're going to get a small local change in membrane potential that will influence the cell. And so here, I just want to go back and remind you that a couple of classes ago, we talked about neurotransmitter systems. And remember, I told you I'm not super concerned about the specific neurotransmitters and what they do right now, right? Like what I want you to remember is that some of these neurotransmitters primarily create excitation. They make it more likely for a neuron to have an action potential. Some of them create inhibition, they make it less likely to do that, and some of them it depends on the receptors that they bind to. And so what we're about to talk about is exactly this, right? We're going to say, well, like what is actually happening to create excitation and what is actually happening to create inhibition and what does that look like in the cell? Okay. So one thing that I will tell you is in order for a cell to create an action potential, and I know that you don't really know what that is yet, right? But like hopefully you at least know that word from general psych, right? We're trying to create an action potential. We have to get the membrane potential to a specific threshold. Usually that threshold is like -55, -60. It's different in different textbooks because it's different in different contexts. But it's less than the -70, right? -70 is resting potential. So we have to change the membrane potential. So if a neurotransmitter is released and that neurotransmitter binds to a receptor on a sodium ion channel and it opens that sodium ion channel, sodium is going to come into the cell. So this is what we mean by sodium influx, right? Influx is in, efflux is out. When that sodium comes into the cell, it is going to decrease the membrane potential. So we started at -70, right? That's resting potential. And we say that the membrane is polarized, right, at -70. If positive ions come into a negative environment, this is just like a number line. So I actually encourage people, if they're struggling with this, like come up with your number line. We have 0, right? We are at -70 for our resting potential. If we have sodium ions come in, that is going to make the difference between the inside and the outside of the cell smaller because you are going to decrease that membrane potential. And we call that depolarization. The inside of the cell has become more positive than it was at resting potential. It is not more positive than the outside of the cell. So that's not, it has not reversed the polarity. It's just we were at -70, some positive sodium came in. That has made the difference between the inside and the outside of the cell smaller. So it was polarized. We have now depolarized that membrane a little bit. And then what we are going to see next class when we talk about action potential, the more you can depolarize the cell, the closer you get to that threshold to create the action potential. So if you can do something to get sodium to come into the cell, you are going to decrease the membrane potential, you are going to depolarize the cell, you are going to make an action potential more likely. And that's how we defined, right, these neurotransmitters that create excitation. Okay. So show me that you're paying attention. Show me that you're still there. I know we're getting near toward the end, but why is sodium coming into the cell? If a ligand binds to the receptor, why is sodium coming into the cell again? I'm like Duolingo. I keep repeating concepts. Make sure that you got them. So the sodium channels have been closed. A neurotransmitter binds. It opens the channel. We say that sodium, there's influx, but we miss some information here that we covered before. Why is there influx?
The sodium is at a lower concentration inside, so it wants to move to that concentration.
Exactly. Concentration gradient and the inside of the cell was negative, so
Electrostatic pressure.
electrostatic pressure, it's pulling the positive ion in. Exactly. So this is set up at resting potential, right? We are set up for if you're opening those sodium ion channels that you're going to get depolarization of the cell. And anytime you get sodium to come in, you're going to create these EPSPs. All right, the IPSP is different, but just opposite, right? So IPSP could be created in one of two ways. It doesn't have to be both. So sometimes what will happen is potassium ions will leave the cell. So something will happen to open potassium channels. If a potassium channel opens, that disrupts potassium's equilibrium potential, and potassium will leave the cell. Something else that could happen is something could bind to a chloride channel and chloride would come into the cell. Because remember, we said that if a chloride channel opens, the concentration gradient is higher, so it will pull it in. Either one of those things is going to make the membrane potential bigger than it was at rest. Because if you have a negative environment that already has positive potassium in it, and a positive leaves a negative environment, it's going to make that environment even more negative. Or if you have a negative environment and a negative ion comes in, like chloride, it's going to make the inside even more negative. So when we make the inside more negative or less positive than it was at rest, that is hyperpolarization. The cell was polarized at -70. It is now more polarized than it was at rest, right? It has even bigger difference between the inside and the outside of the membrane. And that is pulling the threshold, that is pulling the potential away from the threshold that you need for action potential. So that's going to create inhibition. That's going to make it less likely for the cell to fire or have an action potential. Okay. So when you're at the level of the cell body and the dendrite, and a ligand binds, a neurotransmitter, a drug, a hormone, and it changes these ion channels. It opens these ion channels. Which ions enter or leave the cell is going to change the membrane potential. It is always, when you talk about excitatory, we are almost always talking about sodium, especially for this class, right? That is just always sodium is coming in to depolarize the cell. But if potassium is leaving or if chloride is coming in, you will hyperpolarize the cell, and that's going to influence the likelihood that the postsynaptic cell will fire. Okay. Questions before we continue? Okay. We got one more thing to talk about then. So each of those we're thinking about as like one synaptic connection, right? One place where a presynaptic cell releases a neurotransmitter and it binds to a receptor on a postsynaptic cell. But again, that is not what is happening, right? In the real world, you have your postsynaptic cell and it has multiple presynaptic inputs that are coming. So again, right, this is student A sitting in the middle of the classroom, and here is student B, and here is student C, and here is student D, and they're all giving different candy, right? They're all giving different information to the postsynaptic cell. That postsynaptic cell is going to have to integrate that information and sum that information in order to change the membrane potential at this area called the axon hillock. So the axon hillock structurally is the area that connects the cell body to the axon of the cell. And we are trying to change that membrane potential, like we said, to a threshold that will trigger an action potential. And so what's happening is the cell is summing that information. So the song that I played when you came in, that's Violent Femmes, Add It Up, which is one of my favorite songs. And I chose it because of this like add it up, right? Like you gotta add it up, you gotta add it up. That's what the cell is doing. It's summating this information. And there are two different types of summation that the cell is doing. So the first type is called spatial summation. And spatial summation is the postsynaptic cells are summing information from multiple presynaptic inputs across different locations on the cell at the same time. And I will explain this image here in a second. I know this image is confusing, but let you write down what I have up here. Okay, so in this image, we have the postsynaptic cell, and then we have multiple presynaptic inputs. And what we're doing is we are summing, right, across space, assuming that all of these are giving information at the same time. And so what these figures here on the bottom are, and I'll try to, oops, sorry, that was too much. Zoom this in. It's saying that if you were only considering input A, right? If you are thinking about presynaptic terminal A releases a neurotransmitter that opens a sodium channel and a little bit of sodium comes in, that is going to change your threshold, or sorry, your membrane potential a little bit. So when we talk about membrane potential, we often graph it. So you have the membrane potential here over time. And so this is saying, hey, this part of the cell was hanging out at -70, and then a little bit of sodium came in, which decreased the membrane potential, right? Depolarized the cell membrane just a little bit, but then it dissipated and it went away and we came back down to -70. And then this is saying B is doing the same thing, right? So B is releasing a neurotransmitter that is opening a sodium channel and it's depolarizing that part of the membrane a little bit. Either one of those on their own are not going to have a big impact on the cell, right? The cell is going to just get a little bit like, oh, I just got a little depolarized in this area, right? Just like a little bit. But if you sum those two, if they happen at the same time and you sum those two, then you're going to get a bigger change in membrane potential, right? So you're going to get a bigger depolarization. So if neuron A needs, this is very arbitrary, but if it needs 5 Hershey kisses to reach the threshold to get to an action potential, then, or sorry, the postsynaptic neuron, then if neuron A is only giving 1 and neuron B is only giving 1, any one of those on their own is not going to make enough of a change in the postsynaptic cell to get it to action potential. But if you sum neuron A and neuron B, now you have 2 Hershey kisses at the same time, right? And if you had 5 or presynaptic inputs and they all were summed at the same time, then you could get to that 5, right? And then you could get to that threshold. The same thing can happen if you have an IPSP. So I think we used in class, like, let's say it's like the almond Hershey kiss, right? The less likely to be liked, right? To get the one with the almond in it. So let's say you get that and you're like, oh, that's a hyperpolarization. So if neuron C is hyperpolarizing you a little bit, and neuron D is hyperpolarizing you a little bit, and those happen at the same time, you're going to have a big hyperpolarization, right? You're going to have a greater hyperpolarization, and you can sum those up. What is happening in this last figure here? What is the ultimate change in membrane potential that we're seeing here? Is there any change?
Cancel each other out.
They cancel each other out, exactly, right? Because you're summing those. So if A is creating depolarization and C is creating hyperpolarization, you're getting nothing, right? They're going to cancel each other out. You're going to have no net change in the membrane potential. So it's all about what the cell is adding up. Okay. We can do the same thing with temporal summation. So temporal means over time, right? So that word means time. So now we're not considering across space, we're considering over time. So we're only thinking about one synaptic connection, right? So let's say A to the postsynaptic cell, but we're thinking about it over time. Temporal summation is when you can sum that information because there's been a really rapid exchange of neurotransmitter information. So this, if we're using the Hershey Kiss, right? If this is, I'm just going to use like the two names that I see up here right now, which are Callie and Xander, right? So let's say Callie is talking to Xander, and Callie is the presynaptic cell, and Xander is the postsynaptic cell, right? If you need those 5 Hershey Kisses to get to a critical threshold, if Callie gives Xander 1 Hershey Kiss, and then just waits a second, Xander will be like, yum, Hershey Kiss, and eat it, and then it'll go away, right? Like that change will dissipate. And then Callie gives another Hershey Kiss, right? But like the same thing will happen. So what needs to happen is Callie needs to like peg those Hershey Kisses at Xander, just like one right after the other, right? In really rapid succession so that they can't dissipate, so that Xander can't like eat them and make them go away, right? And Xander will collect them and will build them up. And that can build to depolarize to a critical threshold. And then the same is true of IPSPs as well, right? If you are creating an IPSP and you create one, some chloride comes in, you hyperpolarize, but then the cell can restore back to its membrane potential, and then another one happens, you're not going to have a big change on the cell. But if many IPSPs happen in rapid succession, then the cell is going to integrate that. The cell is going to sum that. Okay, so questions about either of those, questions about temporal summation, spatial summation, what we're talking about here. Okay. So it's really important to remember that the postsynaptic cell is not just summing information spatially, right? Or is not just summing information temporally. All of these things are happening at the same time, right? So like Xander might be getting a lot of information from Callie that is excitatory and might be getting some information more slowly from McKenna. I'm just using names that are up on my screen right now. That's inhibitory, right? And so Xander's just constantly, right? And then like Abby might come in with more excitatory, right? So Xander's just constantly, right, balancing these things. And I'm trying to find where I had, here we go. And so the membrane potential at this part of the cell, even though we talk about resting potential and we talk about that baseline, that membrane potential is always fluctuating. It's what we call graded. So those potentials near the dendrites in the body are just constantly varying in size, right? -70 is the baseline, but it'll be like, oh, I'm at -90. Oh, now I'm at -60. Now I'm at -80. Now I'm at -70. Now I'm at -65, right? And it's just like constantly fluctuating until we can get to that critical threshold, which is what we're going to talk about next class. Okay. So I have this video here. I posted this on Canvas just as an extra thing to do in preparation for next week. We're going to watch it in class. So if you don't get a chance to watch it, we will watch it in class. But if you're really struggling with this and you want to think about it over the weekend, this is a really nice visual to show you excitatory and inhibitory postsynaptic potentials, and also to get a sense of what that means when one is being integrated temporally, right? Or two are happening at the same time. And so this is where we're going to pick up on, what's today? Thursday, Tuesday, right? For you all. Hopefully we'll be meeting in person. If not, we'll be back up here. I don't see that happening. But definitely come with questions. I know we don't have a ton of people today, and I know it's been a little bit silent. But I know that not everybody is getting this. So once we're back in person, I need like come with questions, come with places that we need to clarify. And then we're going to kind of move on to talk a little bit about the action potential. All right. What do we need before we log off? Any questions? Any last thoughts? I'll stop moving the Prezi. All right, you all. Thank you for bearing with me in this very awkward format. I appreciate, especially those of you who are willing to speak up and participate a little bit. So I am not holding formal office hours this week because it's not efficient for me just to sit on Zoom and wait for people to log in, and I don't know when they log. It's like I have to pay attention to that. But I am very, very happy to meet. If you just shoot me an email, very happy to set up a Zoom meeting and we can chat about any of this stuff. So. All right, I'm going to do the awkward wave goodbye as people log off. You may log off. I will stick around if you need me later on. Bye, Abby.
Thank you.
Thank you, Xander. See you tomorrow. Bye, Callie.